Okay, so we're gonna try to get started pretty close to time here now that everyone has joined us. Oops. From the waiting room, looks like one person might be having a little bit of issue here. My name is Nicole Scott. I am the project manager for the Agricultural Genome to Phenome Initiative, which is hosting this workshop. We are funded by USDA NIFA on 2 separate awards. You can go to our website, AG2PI.org. That is certainly where you probably registered for this event. And this is also where you will find a recording of this workshop along with our other workshops and field days. Oh, I'm sorry, I clicked the wrong button. There we go. So today's workshop is on crop modeling and looking at the genotype by environment interaction. Our presenter today is Gerrit Hoogenboom. He is preeminent scholar of the Food System Institute and professor of agricultural and biological engineering at the University of Florida. Spent over 30 years of experience in the development and application of dynamic crop simulation models and decision support systems. And he is the coordinator of the decision support system for Agrotechnology Transfer, DSSAT, one of the most widely used crop modeling systems across the world. So with that, Dr. Hoogenboom, if you would like to go ahead and get started.
Okay. So thank you for this kind introduction. I really appreciate the opportunity to talk a little bit about crop modeling with you all this afternoon or this morning, depending on the time zone you're in. So I have a few slides prepared, and then after that I will try somewhat of a live demo with the DSSAT system. So that's a crop modeling system I'm responsible for. So first of all, a little bit of background about agriculture. I'm sure most of you are really in the agricultural business, because otherwise you probably wouldn't be listening to this seminar. So as we all know, that agriculture is really has affected a lot by uncertainty and risk. Farming by itself is really a risky business. It's especially weather that has a big impact on production. We have the day-to-day weather variability. Farmers have to deal with weather extremes, and currently in the US, we currently have a very hot spell right now, and we have areas that are under drought. We also have seasonal variability, and we have spatial variability. On top of that, the economics are also very much uncertain. The prices a farmer might receive for his crops, and also the input costs, with currently the cost for fertilizer going up very rapidly because of the energy cost. Then we have pest and disease outbreaks, and then also last but not least, we also have various countries who have different policies in place to control some of the exports as well as import of agricultural production. So really the question I would like to pose as part of this seminar is, computer models have help with understanding the genotype by environment interactions as part of this whole area of uncertainty and risk within agriculture. And also could computer model predictions help reduce the uncertainty and risk. So for those of you who are familiar with more the traditional economic approach, many of us who work in agriculture in general, we conduct field experiments. We are sometimes referred to these as trial and error, where we have a certain hypothesis where we might be looking at the impact of different fertilizers, impact of different varieties, planting dates, etc. These are replicated experiments normally. We collect data over time, we measure yield, and then hopefully also conduct these experiments at least for multiple years. But we know that even if we conduct experiments for different years, it is highly unlikely that the outcomes of 2 experiments allows us to look at what could happen in the coming weather years. So that's why we want to introduce a little bit of computer models, especially with what we call a systems approach, where we can actually try to predict the system in the future, and where we take the data we've collected as part of the experiments and summarize this information in type of computer models. And really what we want to do in the agricultural system as a whole, and especially when we take agricultural research outcomes and want to provide that information to our stakeholders, first of all, we want to understand the agricultural system. So that's why we conduct field experiments. But really want to go one step further, then we also want to be able to predict if we change some of the inputs or some of the locations, what if the outcomes, those conditions will be. And then when we work with our stakeholders, we really want to look at what we call control and manage. We want to provide our stakeholders, our farmers, with options, and they then ultimately will make a decision which of these options they want to apply in their future agriculture management practices. We sometimes refer to this as options for adaptive management and risk reduction. So with respect to this whole systems approach, really, we first of all want to start with what we call research for understanding. So again, we collect a set of experimental data, and then we develop a model. And that model could be a statistical model, that could be a model based on artificial intelligence, which is becoming very popular. Could even be a mental model. So basically just in our mind, we have a certain hypothesis and we evaluate the hypothesis on the data we collect. Or we can also look at more complex crop simulation models, which I want to introduce in this particular seminar. So with these models, then we can do some type of predictions. And then, of course, we need to evaluate these predictions with real data, preferably from another experiment conducted during second year or data collected in another location. And then we use the test of these predictions to help improve the increased understanding of our overall agricultural system. And then that could lead actually to more research questions. And we know that when we actually conduct agricultural research, the agricultural system is extremely complex. We continuously, in some cases, generate more questions than we actually have answers to our research that we have conducted. Then we also want to look then, take the same information and look at what we call problem solving. So again, we take the same type of models, could be a statistical model, could be an AI model, and integrate that to what we call decision support, and look at application for problem system, for problem solving in the real agricultural system when we work with stakeholders. So then the question I want to pose in this context as part of this seminar, then what is a model? In this case, I want to introduce what we call crop simulation models, where we're trying to integrate the information and the knowledge across the agricultural discipline. But from the outside, agriculture might come across as one single discipline. For those of us who work in the agricultural field, we know that actually there are many sub-disciplines people work on. Be doing research in crop physiology, people might be working on plant breeding, we'll be looking at agronomy in general, looking at the impact of weather, and agrometrology, soil physics, soil chemistry, soil fertility, plant pathology and entomology, and even economics. So again, within the agriculture discipline, there's a whole set of sub-disciplines. So with these crop simulation models, then we want to integrate the best knowledge out of the sub-discipline into a set of mathematical equations that then allows us to do these types of predictions. Again, then ultimately, really what we're after is, first of all, we want to do science for scientific understanding, but we want to integrate these models into decision support systems as well, so that we can provide advisories to managers, stakeholders, all the way from on-farm management to larger scale policy and decision making by our general governments. So that provides us then with different management options we can use for these models. The models never actually tell us what to do, but we can evaluate different options. And then ultimately what we want to get to is provide some type of actionable information for our stakeholders. So what we're looking at here, probably many of you might be familiar with this, is a rice plant. And it's somewhat difficult to see probably on the slide, but you see a lot of boxes, you see some arrows, you see some edges connecting the different boxes together, the different circles together. And these boxes and circles represent different processes that you might see in a rice production system. So this happens to be flooded rice. So we can see that the rice plant is actually planted under flooded conditions. So we have to deal with the simple processes such as photosynthesis, respiration, biomass allocation, with water uptake, transpiration, many of the soil processes, both physical processes and chemical processes. So really, we trying to translate in all these different processes in a set of partial differential equations, and then allows us to predict growth over time, predict development over time, and then ultimately get to yield in some of the yield components. So then what I want to introduce as part of the seminar is the crop modeling system I've been working with as part of our group effort here at the University of Florida, and is actually a collaboration with a whole set of different institutions, which we refer to as the Decision Support System for Agrotechnology Transfer. And again, hopefully we'll be able to show you some of the examples later on after this introductory presentation. So the DSSAT system by itself is actually already quite old. It was started as a project funded by United States Agency for International Development, referred to as the IPSNA project in the 1980s. It ended in the 1990s. And actually the overall goal was to look at a systems approach to help solve food security problems in developing countries. So the original crop models actually were not developed in the US as part of this project. We're looking at food security in developing countries. As part of DSSAT, we brought together different crop models. Some of you might be familiar with the series maize model, with the series wheat model, the sorghum model developed here at the University of Florida, and a whole set of other models. So the outcome of this IPSNA project was actually then the decision support system, DSSAT, which combined the different crop simulation models. Another outcome was what we call a minimum data concept. And again, I'll show you some of that at the end of this presentation, but really to help you as an agricultural researcher define the minimum set of data that are required to run these crop simulation models. And then also as part of that, we try to come up with data standard to allow us to look at compatibility of different crop models to be run with the same set of input systems. So the first version of DSSAT was released in 1986 as part of the IPSNA project. And even when this project ended, the actual development of DSSAT has continued, actually without any funding provided by USAID or any other agencies. We've developed various versions, and as of last year, we're currently working with version 4.8, which can be obtained from our DSSAT portal, www.dsat.net. So these original models were based on original scientific publications. What is shown here on the left is the original series maize book, which was developed by Alan Jones and Jim Canary at Texas A&M and USDA ARS. In the center, it's a little bit difficult to see, but it was a publication on the series wheat model, which was originally funded by CIA to actually predict wheat yield in Russia and other countries in that area. And on the right, you see the original paper for the soybean model published by Gail Wilkerson, Jim Jones, and Ken Booty. Some of the original user guides also for the different individual models. So we had a model for, also as I showed you earlier, for rice called series rice. We had a model for barleys, for wheat. We got a dry bean model and a peanut model, which was all combined in these very old user guides, which are now more or less stored in what we refer to as the DSSAT Museum. Another note of interest that the original models were actually distributed on floppy drives, as you can see here on the left, 5 1/4 inch, and we went to 3 1/2. Then we went to CD, and currently we've moved away from all that media, which is providing you with an electronic download. Also, the original software was sold for $495, and we reduced the price. And then finally, we also made it available as a free download. And also on top of that, currently the source code is completely open source using the 3-class BSD license, which can be obtained from the GitHub portal again. So when we want to talk about DSSAT, it's not only a crop modeling system, which is what I sometimes refer to as a crop modeling ecosystem. And really what I'm trying to refer to here is that we first of all has a whole ecosystem of people. We have tens of thousands of users actually across the globe for using DSSAT. We also have a whole group of trainers, not just at the University of Florida, but also in different countries, who are actually training other people on how to use these crop models. And then we have a group of what we call crop model developers, who are working with us to advance this crop modeling system. So within the DSSAT crop modeling system, we have models for not only different food crops, but also for feed crops, for animals. We have models for fiber crops, for biofuel, and those for vegetable crops as well. In order to work with these crop models, it requires tools to prepare the data. It also requires utilities to actually analyze outcomes of these crop simulation models. As I already mentioned earlier, the outcome of DSSAT was also that we now have a minimum data set, which we defined for what we call model calibration, to fine-tune some of the parameters, and then also for model evaluation. And then we also have a set of data standards that allows people in different areas to use the same set of data for model applications. So as I said, we have quite a few users. As of a couple of weeks ago, at least the number of people who've requested a copy of DSSAT version 4.7 and higher is over 17,000. And what you see here is the distribution of the modelers across the globe. We have a very large modeling community in India, in China, as well as in the United States, in Pakistan and Brazil. But we are over 184 countries where this DSSAT system is currently being used for a whole range of applications. So this is the DSSAT shell, and again, hopefully at the end of this presentation, I will be able to provide you with a little bit of a demo of what the software does and what are some of the opportunities to look at the G by E and even by M interactions with existing data set we have available in the DSSAT shell. And actually, DSSAT by itself, what we refer to is just a shell, because underneath the shell, we actually have what we call a cropping model, cropping system model. We refer to as the CSM model. This is integrated, what I showed you in the previous slide, as the series maize model, the series wheat models, the original soy grow model, peanut grow model, and a whole range of other models. And actually, you can take this model out of the shell, again with the source code being available on GitHub, and you can run it on different platforms as a standalone crop simulation models. But for those of you who are not very comfortable with taking source code and compiling it, you can actually then use it within the shell. We provide you with a set of support software that allows you to enter weather data, that allows you to enter soil data, crop management data, and then the various tools available to help you estimate some of the parameters. Once the model has been calibrated and evaluated, then we have a range of applications, looking at single season simulations, looking at crop rotations, looking at spatial applications, looking at in-season yield forecasts, as well as climate change. And underneath that, for those of you who are already working with DSSAT or will be working with DSSAT, we actually provide you with a whole set of databases with existing experiments that have been used to develop the DSSAT crop modeling system, that also have been used for crop model calibration and crop model evaluation, including weather data, soil data, real experimental data, and observed data. So within that system, then we have to deal with what we call input files and output files, all linked to the crop model. And hopefully when I run a demo, I will be able to show you some of these examples. So we have the experimental data files, we have weather files, we have soil data files. All of these are actually currently in what we call text format or ASCII format. So we're not trying to use our databases, because in the past we found there are quite a few limitations using databases. And then the genetics, I will get back to in a minute as well. Then when we run the simulations, it creates a whole set of output files. We have what we refer to as daily output files, where each individual time step, normally a daily time step, the model creates an output which is stored in these output files. And then we also have summary output files, where we store the summary of these simulations in another set of output files. So with respect to the structure of the model that is shown here, so we have the main program on the left-hand side. We have in the center what we refer to as a land module. And we have a primary set of modules that deal with the weather data, that deals with crop management, that deals with the soil-plant atmosphere interactions with the soil and with the plant. So you might be wondering why we have one crop system model, while earlier presentation, part of my presentation, I showed you individual models. So as you really look at a cropping system, if you look at, for instance, soil water flow and soil chemistry, it doesn't really matter what type of crop is being grown in a particular piece of land. So already in the early days of the DSSAT simulation, we decided that we needed the same set of computer code to simulate soil water dynamics, soil nitrogen dynamics, soil organic carbon dynamics, soil organic nitrogen dynamics. So basically we combined the various components of the crop models into one larger cropping system model where we can share the code. And then with this, what we refer to as modular approach, we can also then actually simulate or introduce different modules with different components in a more complex way. So for instance, for evapotranspiration currently, we use a very simple Priestley-Taylor approach if you only have air temperature and solar radiation data available to help you estimate potentially T, but if you happen to have wind speed, relative humidity, we actually also have a second option we refer to as the FEO56 Feynman-Monteith equation, and to help you estimate potentially T. For organic matter simulation, which is quite important currently when we talk about soil health, we have a simple model we refer to as the series organic matter module, shown here in the bottom part. But we also introduced the Sentry option, where we actually converted the early version of the Sentry model into a daily time-step version that allows us to do a more comprehensive simulation of the soil organic carbon and soil organic nitrogen balance. Then last but not least, we have, with respect to the plant, different options available to introduce different plant modules. So you can see here, basically in the center part of the panel, we still have the original series maize code, which has been changed over time to deal with the simulation of development and growth of a maize crop. But in parallel to that, we also have introduced another maize module we refer to as the series X maize model. So as a user, you can actually decide if you want to run series maize, the series X model, and compare the two different options. The same has happened to wheat simulation, where we currently have 3 different modules. For sugarcane, we have 3 different modules. So depending on the crop we're working with, we have different types of modules available. And we're currently still adding new modules to the DSSAT system as well because of this modular approach. Now on the left, you see what we refer to as the crop grow template. So it originated from the grain legume model for soybean called soy grow, as well as for the peanut grow model. Over time, we first of all developed a generic grain legume model that allows you then to simulate cowpea, dry bean, chickpea, and a bunch of other crops. We also found that this crop grow template is actually quite robust and allows us then also, when we turn off the nitrogen fixation for the grain legumes, actually simulate a tomato crop or also even a cotton crop. So we are slowly adding more crops, also using the crop grow template as well in this whole system. And then the crop grow template was also used a couple of, about 10 years ago, to develop what we now refer to as a pioneer forage model, shown here in the center part of the panel, that allows us to do long-term simulation of an alfalfa crop, where we can actually look at mowing or harvesting, multiple harvests of an alfalfa crop, a Bermuda grass, or a brachiaria grass. And again, we're adding more forages to our crop modeling system as we speak.
Sorry to interrupt, but before you move on, did you see the question in the chat about nitrogen transformations and leaching?
No, I didn't, sorry.
Okay, so Rajiv is asking about nitrogen transformation and leaching. Is there any module to estimate these?
Yes, okay, now I see the chat. So if we go back here, so we have a lot of modules. So the overall cropping system model does deal with both the inorganic part of the nitrogen balance and the organic balance of the nitrogen balance as well. So we have a one-dimensional soil structure, so we can move water both vertically down as well as up. And with that, we can also look at the different nitrogen transformations. So within our crop model, we deal with nitrate, we deal with ammonia, and we deal with urea in the system. So if we have drainage of water occurring at the same time, we're also leaching nitrogen to the groundwater as well. And hopefully I will be able to show you some examples of that later in my demonstration as well. So then I get back to the cropping system model. So you already saw part of this slide earlier in a slightly different format. So in order to run these crop simulation models, you first of all need daily weather data. So we need the minimum daily total precipitation, we need daily total solar radiation, maximum and minimum temperature. To go back to the nitrogen question as well, we do simulate a one-dimensional soil conditions, so we need to have, as a result of that, a profile information by soil layer or preferably soil horizon. We need to know the soil physical characteristics as well as soil chemical characteristics of each individual layer. We need to know a little bit about the surface conditions, such as slope and color. We need a little bit to know about the drainage conditions as well. We need to know then, of course, management. We need to know planting date, we need to know spacing, we need to know the inputs. So we need to know when the inputs were applied, how much was applied, and the type of inputs that were applied. And then we also need to deal with genetics at different levels. And again, I will get back to the genetics in a minute. So then these models operate in most cases at a daily time step. So we would start our crop simulation models at a planting date or a transplanting date, depending on the crop we work with. So we read the daily weather data in, and then we calculate on a daily basis plant development as shown here. So the progression toward flowering, the progression towards first seed, and towards maturity. At the same time, we would calculate photosynthesis, would calculate transpiration, biomass allocation of the different plant components. And then ultimately, we would predict yield. In some of the crop models, we actually operate at an hourly time step as well, especially when we look at plant development, because we know some of these processes are quite sensitive to temperature. And even if the soil the photosynthesis processes as well, especially in the crop grow module, which I talked about earlier. But in addition to predicting yield, within the DSSAT system, it's not only about yield, and also when we talk about the farming system, it's also about economics. So we have a simple way of actually calculating what we call net income or gross margin, where we can assign the cost to the various inputs, and we can assign prices to both the main harvest product as well as the byproduct. So then that allows us to calculate net income. But we can also look at resource use. So we know that if we look at an irrigated system, water is required. So we can look at the response of the amount of irrigation required to yield. So we can reduce the amount of irrigation applied, or we can increase, then determine if there's actually a yield response. And to go back to the nitrogen leaching question as well, we can also look then at the environmental impact. So depending on the amount of irrigation we apply, the amount of fertilizer we apply, we can then also look at nitrogen leaching. And also in our current system, we recently added, for instance, also greenhouse gas emissions as well. So then to summarize, in order to run these crop simulation, we need inputs. So we need field and soil characteristics, the weather data already talked about, cultivar characteristics and management. We do need to calibrate these models for local varieties. A lot of times I get questions when people download DSSATs with respect to why is my variety or my hybrid not included in the DSSAT system. And then to be honest, we know that in the breeding community, new varieties are released continuously So this has been one of our biggest challenges really as a user, that we really need to calibrate these models for local varieties, or local hybrids, and that requires some type of observed data. We also recommend that any modeling system, including the DSSAT system, that you try to evaluate your model with what we call an independent data set. It basically is a data set that was not used for model calibration. And then at least then it will show you that the model will perform well. And then you can go to the next step and provide a recommendation using what-if type of experiments with our computer simulations. And in some cases, when we talk about simulations and even talk about artificial intelligence, people work in more practical agriculture are concerned that the whole computer systems are going to take over agricultural research. And at least from a crop modeling perspective, we continuously find data gaps and knowledge gaps. So at least from our perspective, we can always recommend additional experiments that are required to help improve our crop simulation models and to help improve responses of our simulations to certain inputs. So we can do that on the field experiments, or we can do that under a controlled experiment as shown here as one of the SPAR units we had here at the University of Florida quite a few years ago. So what does these evaluations then look like? So this is an example that probably not everyone will be able to collect with respect to the experimental data. But what you see here on the x-axis is day of year, and we used the concept that January 1 is day 1. And for regular years, that's 365 days in a year. So if you were simulated to December 31st, it would be day of year 365. So that's shown here on the x-axis. On the y-axis, you see biomass or dry weight. This is a soybean experiment that was actually conducted at the University of Florida quite a few years ago by one of my colleagues, Professor Hoogenboom. This was an irrigated experiment and a rain-fed experiment. So you see actually 4 lines. The top 2 lines correspond to above ground biomass for irrigated conditions and rain-fed conditions. And the bottom 2 lines correspond to yield or seed growth over time, again for irrigated conditions and rain-fed conditions. So you can see that somewhere around day 230, the rains actually stopped. So you can see that for the green line, which is the rain-fed conditions, you actually decrease in the simulated data, which is the continuous line, while the irrigated experiment continued to increase. We can also see that for yield, there's a significant difference between the rain-fed conditions and the observed conditions. So the data points correspond to the observed data points. So in this particular case, in the experiment, Dr. Budi and probably his graduate students went out to the field every 2 weeks or 10 days, collected a biomass sample, preferably for 1 meter of row, took it into the laboratory, separated leaves, stems, grain weighed, dried it, and weighed it again. And then that allows that type of data to use for model evaluation or model calibration. So again, the simulated line are based on a dynamic simulation using daily weather data as input, using crop management as input. Some cases people think that we're doing curve fitting, but this is truly a dynamic simulation, and you can see that in this case the model responds very well to the rain-fed conditions, where we can see immediate drop in the simulated biomass and also with grain yield as well. To show you some other examples of less data intensity, so many of you are familiar with statewide variety testing programs. In this particular case, I'm using some examples collected at the University of Georgia. Georgia is a quite important state where peanuts are being grown. So in this case, and again, this is a somewhat older data set, using a variety called Georgia Green. What you see here on the map is actually the state of Georgia, and you see 3 locations, Plains, Midville, and Tifton, where these varieties are being tested under irrigated conditions. And again, the data sets are quite old, referring to 1995, 96, and 2001 for Plains, 94 for Tifton, 96 for Midville. And in this particular case, we actually only had yield and yield components. We had flowering date and we had maturity date available for determining the cultivar coefficients for this particular variety. But that results were quite encouraging. So you see here the one-to-one graph of final yield, simulated yield on the x-axis, measured yield on the y-axis. So the root mean square error for these calculations was about 78 kilograms per hectare. So the model did a very good job. But again, this was calibration. So we modified the cultivar coefficients so that we more or less had an optimum fit when we compare the simulated data to observed data. However, when we take this data set then into farmers' fields, and again, this is an older data set for 2003, you see 3 different fields here in South Georgia. And what you see here is the simulated pod yield over time. So the blue line is what we simulate. And you can see here that we had some destructive samples collected. And again, the model did a fairly good job in being able to simulate the yields from this farmer's field of 3 different locations. So this gives us confidence that actually after calibration, evaluating the model with this independent data set, the model does do a very good job in being able to simulate these conditions. So some other examples. What we see here is again some data from Georgia variety trials. In this case, is for soybean. Again, these varieties are being evaluated for multiple locations, multiple years, as I showed you in the earlier slide for peanut. In this case, we had more environments available for soybeans. So what is shown here on the left is actually the average rainfall observed for each individual growing season for one variety that is being evaluated. And on the y-axis, we see yield. And we can then see carefully, if we look at response here, in order to grow soybean in the state of Georgia, that probably we need at least between 2.5 or 3 millimeters per day to get a reasonable crop. And if we get rainfall above about 4 millimeters per day, we actually don't see any response to the yield for these variety trials. So on the left, we see purely the response on observed data. But then we took the same daily weather data, soil profile data, and the genetics from these locations and entered that into the crop model. And what you see here on the right is actually the response of the crop model. And you can see that the model shows the same response as we found under the observed yield data. So this is for temperature. And we can do the same for, precipitation, we can do the same for temperature. So here on the x-axis, we have the maximum average temperature. We can actually see that for the distance in the state of Georgia, the higher the maximum temperature, the lower the yield. So soybean is not necessarily liking the high temperature in this particular state. With again on the left part the observed yield data, and on the right we see the simulated data as well. So again, the model does do a very good job in predicting these responses. So, so one of the questions in the chat is on how many years of research data is recommended for appropriate simulations. So if we purely talk about model calibration, we recommend that you have at least 2 environments, preferably 2 different years or 2 different locations for calibration of your, especially cultivar parameters. And then preferably another environment, maybe another year or another location for model evaluation. So the data I showed you from these variety trials is really where we actually have more data available, and I'm really trying to just demonstrate the application of what we can do if we have a larger set of data, and to show you how well these models respond. So with respect to crop model applications, my title is really looking at G by E by M interactions. But if you look at these interactions, we can use these models for a whole range of applications. So the original crop models, when they were introduced as part of the USAID project, was really looking at yield gap analysis in developing countries. We can look then extend that to precision agriculture, where we can look at yield variability even within a field, and also try to understand why we have yield differences, and then use that information for prescription recommendations, again using these crop simulation models, assuming that you have accurate information available across space for particular fields. You can use these models to look at irrigation management. So as I showed you earlier, we could look at the amount of water required for irrigation management decision. We can also look at when to apply water and how much water to apply as well. We can turn that around and then also use these same models to look at water use projection. Again, I don't have the example in this slide set, but when I was a faculty member at the University of Georgia, we were actually tasked to project how much water would be required for irrigated agriculture in the state of Georgia in 2030 and 2050 in the future. We can then also take extending from irrigation to fertilizer management. We can look at soil fertility management, look at recommended inputs for fertilizer, when to apply fertilizer, and how much fertilizer to apply. We can look at plant breeding and the genotype by environment interactions, and maybe I can show you some examples later on, nice examples. Yield prediction for crop management. And then also we can look at of course, climate change applications becoming quite of a challenge. With projections, we have the GCM projections showing how climate will change in 2050 and beyond. But then we're really interested also to see what the impact would be in agricultural production. So again, these crop simulation models are one of the unique tools to help us look at climate change, uncertainty, adaptation practices, mitigation, etc. We can look at climate variability as part of those applications too. We look at soil carbon sequestration, environmental impact, land use change analysis, targeting aids, early warning systems. We have several applications currently where we're evaluating these models in Africa to make predictions in season and provide us that information to governments. We have some examples again in Ethiopia. And we've used these models to look at biofuel production. So it's not only about feed and food, but it also could be at energy, and then risk insurance and rainfall analysis as well. So so dig a little bit further then into the genetics, because that's part of the seminar that many people are interested in. So I talked earlier a little bit about these genetic coefficients, and I also talked to you about this generic crop module we have called crop growth. So we're basically trying to use the same set of equations to predict growth, development, and then yield for different types of crops. So the way we handle that is what we refer to as a set of species parameters and functions. So these type of parameters really define the temperature response of a particular crop with respect to development, with respect to photosynthesis, response of the crop to solar radiation, to photoperiod, to CO2, composition of the plant, different plant components, and a whole set of other parameters. And these genetic coefficients are actually stored in an external data file. So as a user, you can actually access the data file and to see how we parameterize these coefficients for the different crops, which we simulate with this crop growth module within the DSSAT system. But we also have, on top of that, what we call ecotype coefficients. So we might have different types of groups of cultivars. So for those who are familiar with soybean, we have different maturity groups, where the maturity groups correspond to photoperiod sensitivity, ranging all the way from no photoperiod sensitivity, so 000, to maturity groups 9, where the cultivar is highly sensitive to photoperiods. So we define these as ecotypes. So we have a set of coefficients to deal with that. Then last but not least, we also have cultivar coefficients that allows us then to define the number of photothermal days, of photothermal time to flowering, to maturity, sensitivity to photoperiod, seed size, and a whole set of other parameters as well. So, so again, a question in the chat is, can these models then predict morphological traits? Yes. I showed you earlier in my slide, I showed you biomass, but at the same time, these models also can predict lever index. They also predict the daily crop growth rates, etc. So yes, in order to conduct these simulations, these are all part of the dynamic calculations internally in the crop model. So then to show you an example of the difference between what we refer to as the species coefficients and the cultivar coefficients. So on the left, you see what we sometimes refer to as a temperature response curve. So many of you are familiar with growing degree days, where you have a base temperature and an optimum temperature. In many of these crop models, actually, we like to use what is referred to as a cardinal temperature function. But again, we do define a base temperature. But then we also have two optimum temperatures, shown here as optimum 1 and optimum 2. And also we know that on high temperatures, actually, the crops do not develop whatsoever. So we also have a maximum temperature defined in many of our crop modeling systems as well. So these are species coefficients. So these are defined for each individual crop, especially for the grain legume models and the crop growth models in the species file. On the right, you see some coefficients that deal with photoperiod sensitivity. For many of you, again, are familiar with what we refer to as a critical short day length, which we define for each unique variety. And we also might have a photoperiod sensitivity as well, which we define to as a ppsen.
Okay, so we actually have a few more questions in the chat that I wonder if you could answer before moving on. So the first is, if you don't have the right equipment on the field to measure solar radiation, can you recommend a source online where someone could download it?
Yeah, okay. Yes, so there are various ways to estimate solar radiation. So one way, which I personally don't prefer, but there are equations published in the literature where people just use the latitude and clear sky radiation, which you can actually calculate, Tmin and Tmax temperature and precipitation to estimate solar radiation. But the source we currently really recommend is what we refer to as NASA Power. This is a gridded data set, provides you with daily solar radiation at a half-degree grid. It can be downloaded from the NASA Power site. And in fact, if you go to our DSAT portal, we provide you with a link to this NASA Power data set, and it's updated on a daily basis. And we've actually conducted some comparisons with ground stations, and we find that it is quite accurate.
Okay, thank you. Another question is, does the model consider uncertainties associated with model structure, parameters, and inputs in simulating outputs, such as grain yield?
No, but that's a very good question, but these are purely deterministic models. These are not stochastic models. So if you provide it with the same set of inputs, you get the same set of output. But we do realize uncertainties associated with the inherent equations we have in the model, as well as also especially some of these genetic parameters as well. So we currently don't actually deal with that. We are trying to come up with solutions with the uncertainty associated with the genetic parameters, but again, currently we provide a fixed yield.
Okay, and one more question. There are several here, but I think maybe we can wait until a little bit further into the demonstration to answer some of these. But the last one I wanted to ask was, how does the model differentiate one irrigation system to another, furrow to sprinkler, for example, in terms of water application?
That's a good question, and actually the irrigation routine is not extremely sophisticated. So we basically look at the net infiltration of water into the soil system. So we have what we call an efficiency factor that you need to determine. So if you know the amount of water that's being pumped, you actually need to know how much water actually reaches the soil surface. So if you have subsurface drip, you know that we have 100% efficiency, but if you have an overhead sprinkler system, the efficiency might only be 75 or 85%. So you can determine that as an input into the crop model system. So, okay. So should I go on?
Yes, let's go ahead and go on.
Okay. So, and again, it goes a little bit back to one of the questions about uncertainty. So one of the real challenges with these crop simulation model really is what we refer to as these genotype specific parameters. We really want to be able to be as accurate as possible to simulate the response of different genotypes to environmental input and crop management, but that is still very challenging. So what I showed you earlier with these cultivar coefficients, they more or less emulate certain parts of the crop system, but they don't really directly link to the genetics. So one thing we try to do is actually bridging the gap between biotechnology, breeding, and crop management. And already quite a few years ago, I worked with a colleague who was then based at CIAT, one of the international research centers in dry beans, where we developed a gene-based model as an outcome of the bean grow model, where we actually had 7 real genes, which we then linked to these cultivar coefficients. And that resulted then into a gene-based model. So if a person was able to identify the genes associated with a particular variety, that can be defined as an input into the crop modeling system. So at that time, there were not, as we know, there are no direct genes that control yield, but there are quite a few genes that deal with photoperiod response, can determine indeterminacy versus determinacy, early flowering, maturity, and also seed size. So for dry beans at the time, we have a PPD gene, we had an HR genes, and hence the effect of photoperiod sensitivity, and indeterminate versus determinate gene for fin, which also is present in other crops as well. Early flowering defined as FD, and then various genes associated with seed size. So the underlying idea of this approach, and it's still relevant today, although this was done quite a few years ago, but if you have a particular cultivar, and it's shown here, you see some cultivars for dry bean, red cloud, and Kalima. And if you know the gene map of that particular cultivar, you can actually then define that this particular gene is dominant or recessive, shown here through zeros and 1. And you can use that as a direct input in the crop modeling system rather than having you estimate some of these cultivar coefficients. So then we try to use that as an input into the crop model. So actually, rather than going straight, because we still had these cultivar coefficients, we did try to link these genes to the cultivar coefficients in the crop simulation model. So as an example right here, the current cultivar coefficient that we have within the common bean is what we refer to as the physiological time from emergence to flowering, EM to FL. Then we use 2 of these genes, the fin gene and the FD genes. And we use some simple linear regression to come up with the definition and calculation of the EM to FL parameter based on the presence or absence of these genes. Did the same for photoperiod sensitivity, shown here on the bottom with ppSem. So I showed you this in the earlier slide as well. So again, we use the PPD and the HR genes here to show the relationship between photoperiod sensitivity and the presence or absence of this particular gene and activity of the genes. So again, the R squares are not extremely high, but they were significant. So then we went through various versions of this dry bean model. So we actually looked at 46 different cultivars, which are grown in a whole set of trials across the US, Mexico, Colombia, and Canada. We used some of these data for calibration. So you can see here we have 10 trials with 177 observations for calibration for these gene coefficients, and then 26 trials for evaluation. Then we compare the original crop grow model versus the gene grow model. So the number of days to emphasis shown here is the root mean square error. The crop grow model was still slightly better than for the gene grow model. Days to maturity was also slightly better. But again, keep in mind that for the crop grow model, we actually had to calibrate each individual cultivar separately But for the gene grow model, once we knew the presence or absence of a particular gene, all we had to define was the zeros and the ones in the input file. So the inputs required for the gene grow model are a lot less than for the crop grow model. So these results are actually quite encouraging. For grain yield, actually, again, the root mean square error was only slightly higher for the gene grow model, although we didn't have any particular genes to deal with yield. Above ground biomass was slightly better. And then unit grain weight was, again, was high, relatively high. But again, overall, especially for phenology, the gene-based model did a fairly good job. So then we can go one step further and actually use this gene-based model as a potential breeding tool. So again, the data set is quite old. This is an example for the Kellogg Biological Station in Michigan, where we had long-term historical weather data available, in this case 73 years of data, 1930 through 2002. You can see the distribution of the climate data shown here on the right for maximum temperature and for precipitation. So then what we did is we created what we sometimes refer to as idiotypes, where we had the combination of 96 genotypes, looking at the dominant and recessive values of each of those combinations of these 7 genes we had available. We had 128 potential combinations, so that's 2 to the power of 7, but we actually eliminated the recessive genes of PPD and HR. So then we ran that through the crop simulation models, using this for rain-fed conditions, using 73 years of weather data. So then what the type of simulations you can get is shown here. Again, the graph is not very sophisticated, but this is our version of what we refer to as a box and whisker plot, with the median values of 50% distribution shown here with the asterisks. You can see the 25th to 75th percentiles in yellow, and you can see the low ones in the greens and the blues in the top part. So this shows you the different combinations of the genes, and then that allows us to identify which particular gene combination will perform best for this particular location. So in this graph, it's very difficult to determine actually what would be the best value. If we plot this differently, we can plot this with the mean yield versus the variance. So the variance here is shown on the x-axis. Mean yield is shown on the y-axis. So from a farmer's perspective, we want to actually have a very low variance, which when we work with daily weather data, we know that is very challenging, but because again, as I showed you in my second slide, weather varies from season to season. So what we're looking at here, then the decision criteria would be with determine the one with the highest mean yield and the relatively lowest variance. So in this particular case, the variety shown here in the orange box. So with all genes showing dominant, except for seed site number 3 being recessive, actually provided us with the best performing genotype for this particular location. Then we can run these same type of simulations for different locations. So we did it also for Twin Falls, Idaho, for Prosser, Washington, and then we can actually look at the rankings, and we can actually see that for these different locations, or whether it's quite different, we still see that some of these genotypes, especially genotype number 2 and genotype number 9, were in the top 3 rankings for all 3 locations. So the critical genes here determine that FD is quite important, early versus late flowering, as well as the seed size as well. So again, this is a promising application, but again, one of the challenges we still face is the linkage between the individual genes and the cultivar coefficients. So then the other part, which has been done more recently, is looking at some of the data which are an outcome of QTL studies. So a study which was funded by NSF, conducted by some of my colleagues here at the University of Florida, Eduardo Vallejos, Jim Jones, and Ken Buddy. We looked at QTLs again for different common bean, intracombinant inbred lines, which were grown in different environments. And then they use that data to come up with what they refer to as a statistical model. That is basically is a very fixed model, where we basically look at the Tmax and Tmin and day length values, average values across environments, and then look at the interactions between the different QTLs. But actually we want to take this one step further and actually see if we convert these QTL statics models into more dynamic models with the idea of linking this QTL model again as a separate module into the crop growth model, bypassing then the traditional way how we calculate the number of days of flowering, using this on a QTL based model. So in this particular case, then this would be integrated with the crop model. We use again the same daily weather data as input. We use the cultivar characteristics as input, and then we would replace this QTL model calculation with the traditional flowering date model in the system. Again, the underlying idea would be to look at an input file shown here, and rather than using zeros and one in this particular approach, we're using -1s and +1. And what you should see here on the top is not genes, but it now happens to be the QTLs of the different recombinant inbred lines. And on the left, you can see the various recombinant inbred lines. And the parent material is Kalina and Hamata, which is shown on the first two lines. So again, then the idea would be to take this input file and to help define the particular QTLs, which are responsible for the effect of the different recombinant inbred lines. So we developed a more dynamic mixed linear module where we can then actually calculate on a daily basis the progression toward flowering, using then these QTLs, using day length, using solar radiation, using maximum and minimum temperature to come up with these set of equations to help us do these calculations. So the original data set was based on 5 different sites, ranging all the way from 2 locations in Colombia, Palmyra, which was a warmer site, and Propian on a mountain, which was a cooler site. And it included Puerto Rico, it included Florida, Citra, a site here close to Gainesville, and North Dakota, further north for the period was quite important. So then we compare the predicted modeling using the QTL model, and there's various versions here, but what we're interested in is the one, the panel on the left shown under C, and the one on the right shown D. So the original model is where we parameterize each individual recombinant inbred line and determine the cultivar coefficients. And on the left, we use then the gene-based model to predict the number of days to flowering from planting. And we can see that we see a little bit more scatter on the gene-based model. But if you look at the adjusted R-square, it actually accounts and penalizes the original model because you have to estimate more cultivar coefficients. The gene-based model does a much better job compared to the original model. So this shows promise. So then if we compare this again with the observed data, so what you see here on the right is the 5 locations, North Dakota on the top, Propian on the bottom, and we see the time to first flower for the range of the various recombinant inbred lines, shown here with the various graphs on the right, and on the left we see the same with the simulation, with the dynamic crop simulation model. We see that it is not exact, but we're still very close, seeing the same type of distribution with the dynamic recombinant inbred line model versus the observed data. So again, then we can take this type of modeling approach and ask some questions, especially as it relates to climate change. So how will these recombinant inbred lines respond if we increase the temperature by 1 degree, by 2 degrees, by 3 degrees, and by 4 degrees? So at the top, we see the simulated response for the baseline. And again, the different colors represent the different environments. And again, we see different responses based on the locations. So some locations, flowering will occur earlier. In some locations, flowering will occur later, depending on how we actually predict the effect of temperature. Of course, we were ultimately interested in yield. So as I said, we took this dynamic gene-based module and integrated it into the dynamic crop simulation model of DSSAT. And we compare that to the original model. So what you see here on the left is the original model. Again, we see the 5 different locations. Again, this was calibrated with the cultivar coefficients, and we can see on the right how the gene-based model respond. So again, the gene-based model showed a very similar yield distribution for the 5 sites compared to the original model. So using again this QTL approach is very promising, but of course one of the drawbacks of this approach is that it is very specific to this particular data set. So it's not really very much generalizable to a different location.
There's a question in chat about whether the model accommodates the disparities in industrial standards in estimating GDD from planting to flowering and physiological maturity.
I'm not really sure if I understand the industrial disparities in industrial standards. The whole growing degree concept by itself is questionable because it all depends on how you define your base temperature, how you define your optimum temperature, if you separate your Tmin and Tmax. So the various ways to me using degree days. And actually in our crop simulation models, we try to avoid using the pure degree day, growing degree day concept. And especially actually in the crop grow modules, we actually calculate an hourly temperature response to look at what we refer to as photothermal days. So we go beyond the growing degree day concept, because for us it has very limited, it's very challenging, especially also when we look at a climate change response for high temperatures. So probably didn't really address your question directly, but again, unfortunately, I really don't have time to go into a lot of detail on the underlying equations of crop simulation models. We cover some of that in our crop workshops, which we organize on a regular basis, but these takes about 6 days to explain all the details of the crop simulation models. So I want to get back a little bit to then the data to run these crop simulation models. Goes back even to the question about solar radiation and even with respect to irrigation as well. So in order to run crop simulation models, you do require accurate inputs. So we talked a little bit about soil data, we talked a little bit about the weather data, we talked a little bit about genetics, and we talked a little bit about crop management. So one of the challenges for those of you who collect experimental data is really that you might feel that you have to collect too much data to work with these crop simulation models. So we've tried to define a minimum data set. And in our eyes, a minimum data set is a data set that allows us to run the crop simulation model and allows us to provide us with fairly accurate simulations and fairly accurate predictions of yield. But at the same time, we also define a minimum data set, a data set that can be relatively easily collected by you as an experimental scientist. So it's very much a delicate balance with respect to how much data are required to run the simulation model and how easily it would be to obtain the data on the field conditions. Now I know that if you talk to other crop modelers or if you talk to hydrological models, each of these people have a different set of standards, but ultimately all want to get to the same question, that we want to be able to run these models to predict the genotype by environment, by management interaction. Now the whole concept of a minimum data set is nothing new. As part of the earlier IPSNAP project I referred to earlier, they already organized a symposium in the 1980s where they actually tried to define this whole minimum data set. So at least the crop modeling community has been struggling with this crop minimum data set for quite a long time. So already talked a little bit about the weather data. So in order to run this crop simulation model, this is really a minimum we need: maximum, minimum temperature, total precipitation, and total solar radiation, because we do predict photosynthesis, which is driven by solar radiation. And again, based on the earlier question, if you don't have solar radiation, you can estimate it from sunshine hours. If you don't have sunshine hours, then maybe using the NASA power data set would be a good way of coming up with user radiation inputs. So with respect to the soil dynamics, we have a one-dimensional soil profile. So we need to know the water holding characteristics of each individual soil horizon or soil layer. We need to know a little bit about the inherent nitrogen present. We also need to know organic matter that is present, especially if you use conducting simulations for low input systems. It is very important to also define what we call the boundary conditions. So how much water is available in your profile at planting or the start of the simulation, and how much nitrogen is present in your soil profile at the start of simulation as well. Now, with respect to crop management, of course, we need to know the crop, we need to know the cultivar, we need to know when the crop was planted or transplanted, we need to know spacing, row and plant spacing. And also going back to the earlier question about irrigation, we need to know the dates and the amount of irrigation that was applied. And again, going back to the question about flooded irrigation, it is quite a challenge because in many cases people don't really know how much water is being applied on the flood irrigation. So you need to come up with an estimate how much actually water penetrates into the soil system. For fertilizer, we can deal with both the inorganic fertilizer as well as with organic material, especially residues. So we need to know again the dates, the amounts, and the type of fertilizer that is being applied, the organic elements, the amounts. We can look at tillage operations, the dates and the amount of the tillage, and also know the amount, the type of tillage implement, and also chemical applications, although many of our models currently don't really respond to these chemical applications. So this is the minimum amount of data required to run the model. I talked a little bit about evaluation. So in order to evaluate a model, we need at least the minimum of the minimum would be yield. So if we want to understand differences among experiments and we see differences, then we may need to look at the yield components, or we might need to look at difference in dates as well. So those become quite critical. In many cases, when we talk about these crop models, we state that if we're unable to predict the flowering date correctly and we're unable to predict the maturity date correctly, so basically the length of the growing season, we cannot predict the yield and the yield components very well either. So as I said, the minimum would be yield, and we can be looking at yield components, but we also recommend that phenology is quite important as well, especially starting to work with a new variety of cultivar or hybrid to be able to accurately predict the flowering date and the maturity date. If you're interested in looking at other responses of the model, especially looking at how well it responds to inputs such as irrigation or nutrients, so it could be important, it's not required, but you can look at measuring soil moisture at different depths or different locations in the field over time to compare simulated soil moisture for the different layers versus the observed soil moisture. If you're more interested in nutrients, again, the same. You could take some soil measurements, different depths, different locations in the field. You can also look at the composition of your plants as well. And again, then you compare the simulation, predicted simulations with the observed data. So again, go back to the minimum data set for understanding. So the minimum would be yield. Then the next one would be phenology, so especially the number of days to maturity to determine the growing season duration. Number 3 would be the flowering date, and looking at the yield components with respect to seed size and seed number, and then also final biomass to help us calculate the harvest index. So in the minimum data set, we have sometimes referred to as 3 different levels of data for understanding. So level 1 would be basically just to run the model for different applications. Level 2 would be to calibrate a model and evaluate a model. And then also we have a level 3 data, which would be to possibly collect a more extensive data set if we want to add a new crop to our crop modeling system, or we want to look at a new process that we're currently unable to simulate. For instance, salinity is still quite a challenge in our DSSAT system. So we're still looking for a good data set to help us improve response to salinity, as well as to some other soil factors as well. So with respect to DSSAT as well, some opportunities as well as challenges. As I said, in the early days of DSSAT, we actually sold the software, and now it's being made available as a free software, actually open source, for the model as well as for the tools. One of the challenges we also face a little bit is that the actual model itself is developed in Fortran, and many people are unable to program in Fortran. So we're looking at options for what we refer to as mixed language programming. We can actually bring in C++ and some other programming languages into the crop modeling systems. We continuously to expand the type of tools and applications we have available. So in the most recent version of DSSAT, we actually introduced an in-season yield forecasting application. We've talked a lot about climate change, but actually now we've actually introduced, as part of DSSAT shell, some examples of climate change applications as well. For those of you who are more power users, want to run these crop simulation models across space for many different applications, you can actually port the model itself to a Linux environment. You can run it on a high performance computer, or you can also run it on a Mac system. And again, in that case, you do need to recompile the original source code. Looking also currently at, for those who are familiar with computer science, is a terminology of Dockers and containers, so we can actually put it into a wrapper and look at multiple applications of the crops in simulations as well. So one of the challenges we really face is the lack of financial resources to help advance the crop modeling system. In general, many of the funding agencies are more interested in the applications of the software, and not really necessarily interested in helping improve the models. So we're always struggling to add new capabilities to the DSSAT system. But ultimately, again, the advancement of the DSSAT system is driven by you, the user, who's really interested in working with the DSSAT software. And as I showed you earlier, we have quite a few people across the globe who are currently working with DSSAT. And also, as we've seen in our workshops, there's still quite a lot of interest in learning more about the DSSAT software as well. So with respect to, again, new capabilities within DSSAT, recently we've added new crops, safflower, alfalfa, teff, sugar beet, strawberry has recently been added, but it's currently not available yet in version 4.8. Carinat is a new model, again, not available in 4.8. We have available as an in-house version. Said earlier, we're also adding new crop modules for the crop growth perennial model, for perennial forage grasses, was something we added recently. We added a new module for teff, based on the N-wheat model, we refer to as N-wheat teff. And we are working on different processes. So we are looking in-house currently at salinity, looking at plant phosphorus for different crops. We're also looking at potassium, that's in the early stages of development. We're now also looking at predictions of greenhouse gas emissions, although we are still lacking good data for crop model evaluation. We have an in-house version of looking at a 2D model. Again, we're lacking good data for model evaluation as well. One of the aspects I really haven't talked about much is the linkages with and effects by pests and diseases. So that's one of the main challenges with current crop simulation models. But again, using mixed languages as well as using MPI and Dockers and containers, we're now able to couple different pest modules with the crop models, where we can actually exchange information between a pest module and a crop module, where the pest model can tell us the amount of damage, and then we can simulate the damage in the crop model. And we can provide a pest model with the amount of leaf area or leaves or roots that are required by the pest to grow. And then also already showed you a little bit about what we're doing currently with linkages to genetics and genomics. So then finally, we already talked a little bit about the DSAT portal. So I highly recommend to you, if you're interested in learning more about the DSAT system, if you haven't downloaded it yet, I suggest that you request a copy of DSAT version 4.8, which is our most recent release just made available. We also provide various background information on the DSAT portal as well. And if you download the DSAT software, we also provide you with various papers which have been published on DSAT, as well as a whole range of documentation. With respect to model development, we now have regular hackathons where we get together as a group of scientists. Thanks to COVID, this has turned into hybrid sessions where most of the people are online using Zoom. But we also have some in-person meetings as well. So the last hackathon or DSAT development sprint was held at the University of Florida in early January. So on the bottom pictures, you see some of the key model developers of Ken Booty, Shell Porter, Wolfgang Schilia, and some others. Well, in the various frames, you see some people across the globe helping us work on DSAT based in Brazil, in Germany, in Mexico, in Spain, in Thailand, and South Africa. And the next DSAT sprint is scheduled for last week of July at UC Davis. And again, if you're interested in participating, there's no charge. Send me an email, and I'll be glad to send you and share you more information on this DSAT development sprint. So we also do a lot of capacity building and training. So as I mentioned earlier, we had a workshop in May at the University of Georgia where we had about 54 participants. Unfortunately, you do need to pay to participate in these training workshops because it costs us money to actually facilitate these workshops. The next workshop will again be held at the University of Georgia in 2023. In addition, we also have these workshops at various locations across the globe. I personally conducted a workshop in Ethiopia in March, and we currently have some discussions about having a workshop maybe later this year in Germany, and potentially also maybe one in Bangkok in Thailand. So with respect to modeling, of course, they're not perfect. There are always limitations to these models, limitations to data. There was one question about the uncertainty associated with the underlying equations. That's one aspect we need to deal with, but we need to even be more careful about actually the accuracy of these equations. So what you see here is actually a dam in the state of Washington. And prior to me joining the University of Florida, it actually was the Washington State University, where a crack in the dam occurred. It's one put on dam, and the crack of the dam occurred because of an mathematical error in the design of the structure of this dam. We also know that our underlying equations are not always perfect. We try to make them perfect, but unfortunately, once in a while we do find errors in the equations. And also, of course, we try to improve these equations as well. So we definitely need to be careful when we use these models to make sure that they're accurate. And because we have so many equations, it's always very, very, very difficult to make sure that we don't introduce any errors when we develop new code or look even at old code. So with respect to the modeling system itself, with the agricultural system, we know that the agricultural system is also extremely complex as well. I really haven't talked much about differences between potential production and water limited production and nitrogen limited production per se. I talked a little bit about pests, which currently are not part of our current modeling system. But again, it is quite challenging really to do a very good job to be able to simulate all the components of the agricultural system. I only have talked about crops. We also know that livestock plays an important role as well in the overall agricultural system as well. And then finally, which is quite challenging too, is the whole human decision making process. Our current crop simulation models provide us with answers, but then we provided information to farmers with respect to actionable information. But if we provide this to different farmers, they might actually make different decisions based on the same information we provide them. So that's something we really currently are unable to model. But then having said that, with respect to crop models, the questionnaire facts or fiction, of course, I'm biased, but I think they're very good tools. They are a mathematical representation of a real-world system. They require accurate data as input. They also need to be carefully evaluated. But once we've done that, there are many opportunities for application of these models with respect to prediction of yield, with respect to prediction of resource use, look at environmental impact, also look a little bit at the economic returns, and especially these models allows us to simulate a lot of what-if type scenarios on our computer to evaluate different options. As I said earlier, we find when we use these models the continuous knowledge gaps, so they can be used also for research design and analysis to determine what type of priorities are needed for our field research. We also found that these models can be a very good teaching tool. Once in a while, when I look at these requests of the crop models, I could see that they're being used in a class. Because I might get 10 or 20 or 30 requests from a particular location where the justification is that a teacher told them that they needed to use DSSAT as part of the class project. And then last but not least, they also can be used for policy and planning at a higher level by our governments. And we know that more and more governments across the globe are using models and decision support systems to make more informed decisions. So with respect to the future farms, we do know that with remote sensing, with sensors in the field, with artificial intelligence, a lot of data being collected. We are trying to provide this information to our farmers and stakeholders, which is still quite challenged to use all these technologies, because some of these technologies can be quite expensive. And I feel personally, and of course I'm biased, that in concert with remote sensing, other sensors, and artificial intelligence, these crop simulation models also can play an important role in the future farm. So with that, I'll be glad to answer any more questions related to DSSAT proper, and also I'd like to still got about 30 minutes left to show you some quick examples and demonstration of the DSSAT system. So Nicole, I noticed quite a few questions again in the chat. So how do you want me to proceed?
Yeah, sure. I'll just kind of maybe summarize a couple. So there are a couple questions about the training and whether they only occur in the US. I think you maybe touched on that, but if you want to go a little bit deeper into where the trainings are made available.
Yeah. So we always have our training in the United States once a year in May, so that's a set date. And then we also provide training across the globe, but it depends a little bit on the hosting organization. So if someone is interested in hosting a workshop, then we ask them to provide the logistic support for that particular workshop, and also then provide financial resources for the trainers to come to that particular country and conduct the workshop as well. Salaries are not included, but at least we need to pay for travel expenses to conduct these training workshops. So we advertise these workshops normally on our DSSAT portal, but also through our the DSSAT list server. So last year we organized a workshop in Germany, which was hosted by Sento de Seng, one of our former colleagues here from University of Florida, who is now based at Thun. And because of COVID, unfortunately, only people from Germany could attend. But again, we have a new discussion with him to have a broader workshop, hopefully later this year in Germany. And as I said, we're having discussions of potentially having a workshop in Thailand. And other workshops as well. And again, it varies really a lot on the host who's willing to put up a workshop, because it takes time, it takes resources to actually organize these workshops.
Okay, and then a couple more questions just before you get into the demonstration. One person asked whether the model has been used for perennial crops, especially fruit crops, which I believe you also touched on, but maybe review that again. And then can it predict root growth?
Okay, so the perennial crops, especially tree fruit, is a very good question. It's a very challenging question. So the answer is no. Unfortunately, no one has been able to crack that very challenge of being able to predict tree fruit growth over time. Because one of the challenges is, first of all, if you look at, say, for Florida, a citrus crop, an orange crop, or if you look at the state of Washington, an apple crop, in order to develop a model, we need to have very good growth analysis data, very good phenology data, in order to develop these equations that help us predict long-term growth over time. So unfortunately, we have not been able to do that well. We can predict phenology of tree fruit relatively well, but not tree fruit yield. So I did mention perennial crops, but it was mainly for perennial grasses. So we have been able to do that well in our current DSSAT modeling system, but tree fruits currently we don't have in our crop modeling system, unfortunately.
Okay, let's go ahead and go to your next step.
Okay. Okay, so you can see my entire computer screen right now. I hope it's going to work. So, so what I want to show you a little bit of a quick introduction of the DSSAT software. So can you see this, Michelle?
Yes, yep, this looks good.
Okay, very good. Thank you. So basically, if you download the DSSAT software, this is what, and then you install it and you open the shell, this is what it looks like. So the critical part here in the center is what we call the crop selector. So you can click on the different various crop groups we have available. So we have cereals, we have fiber crops, I mentioned the forages, the fruit crops. The only one we currently have is pineapple, and that's not really a tree fruit crop. And we'll be adding strawberries to this as well. We have the grain legumes, all crops, etc. So currently we have over 40 different crops under the DSSAT system. So for any of the crops we currently have on DSSAT, we provide you with at least one set of real experiments for which the model was calibrated or evaluated. So what I want to show you here is, for instance, the maize model. So what you see here now in the panel on the left, or sorry, on the right, see 12 different experiments which were conducted in different locations across the globe. Many of these are quite older experiments. So all these experiments come with the crop management I showed you earlier as a required input. They come with the soil data, and they come with the daily weather data. So as an example, I want to again show you something from the University of Florida, Gainesville. This was actually an experiment that was conducted in 1982, quite a few years ago. This experiment consisted of 6 different treatments, ranging from rain-fed low nitrogen to irrigated high nitrogen. So when we run the crop model, it allows you to select the treatments. So when I run the model, this actually goes quite fast. So what I just did, I actually simulated 6 different treatments. Again, to remind you, the model operates at a daily time step, or in some cases it operates at a hourly time step. So we were able to simulate this very quickly on a regular notebook computer for 6 different treatments. So I run the simulation and the model is actually in the background. You have not seen the model whatsoever. So as I said, the model creates a set of outputs. We have a whole set of output files, and I don't really again have time to discuss all these output files in detail. But what I want to show you is again some time series analysis. I want to go to plan grow that out. And I just want to make sure that Can you still see can you see my graphic screen now, called GBuild? Can you confirm this?
I'm seeing the time series plot.
Yeah, okay, yeah, good. So what you see here is a whole range of variables, and you can see the selected runs. So for instance, on the left is what the model can predict with respect to above ground biomass and some other variables all related to plant growth. So we can, for instance, plot pot weight, tops weight and grain weight. So can you see my graph?
Yep, we can see the graph.
Okay, I just want to make sure this is working. Okay. Okay, I won't ask anymore. So what you see here is very similar to what I showed you on my slide set earlier. So right here we have the simulated, which is the continuous line, and the observed data points observed again, slightly different from soybean, but again with the same principle. The more or less the top 6 lines refer to above ground biomass, and the bottom 6 lines refer to the grain weight simulated over time. You see the points here on the right are a little bit later, because actually the model predicted physiological maturity, and these happen to be the biomass samples that were collected at final harvest. So the model didn't predict all the way to final harvest in this particular date. We have some statistic features included that allows you then to analyze the statistics of the model, how well the model performs. And what we personally like to look at is something called the D-statistic, where value of 1 means the model performs very well, a value of 0 means the model performs very poorly. So we can see for grain weight and tops weight that in general most of our D-statistics is above 0.95, close to 0.98 and 0.99. So the model performs very well, but again, keep in mind that this is a calibrated data set, so we haven't really done any individual evaluations of the crop model. So again, when I introduced this earlier in my presentation, I talked about the challenges of conducting experiments only for 1 year of data. So now what I want to take you to is an application using the same set of experiments. So under these sets, first of all, I mentioned we have this crop selector sitting here on the left. So these are all based on real experiments with real data. So if we go to applications, these are more or less what we refer to as hypothetical applications, where we can run these what if type questions. So these are hypothetical experiments, no observed data. In some cases, we might have long-term real experiment weather data available. In some cases, we might have to use weather generator. So then the question really is, we've conducted this experiment for 1 year at the University of Florida in Gainesville. We're looking at a maize experiment, looking at the interaction between irrigation and fertilizer. But how would this experiment correspond to different weather conditions, especially with long-term weather data? So this is where we bring in what we call a seasonal analysis, where we take the same type of experimental conditions and actually evaluate the performance for long-term historical weather data. So I'm going to run the same set of simulation with the 6 treatments I showed you earlier. But now I'm going to run this for 30 years of historical weather data. It'll take a little bit longer, but not really too long, hopefully. So as you see, again, the simulations are completed. So what I did here is I just conducted 6 treatments. We had 30 years of data. So actually we ran 180 simulations very quickly on my computer. So I can show you a different type of analysis. So here we can now account for the uncertainty of the weather data. Again, we have a whole set of outputs, and I don't really have time to explain and analyze all these outputs. But what I want to show you, a very simplistic way of the response of these individual treatments to long-term historical weather data. So many of you, again, are familiar with the box and whisker plot. There's a similar version of wax and whisker plots. where we show the median value, which is the asterisk 50%. We have the 25th to 75th percentile, which is the yellow bars. We have the high yielding environments, which is blue, 75 to 100. 0 to 25 is the green values. So treatment or scenario number 4 is the high input, high nitrogen, high water. So in general, we can see that the median value is quite high. And we can also see that the maximum value and the minimum values are higher. But if you go to the low input experiments, which are number 1 and number 2, we can see that in certain cases, actually, the model performed quite well. So the only differences are that for these treatments, we varied the historical weather data. We didn't really vary the crop management. So here is where we try to account for the uncertainty of weather, while we keep the crop management the same. And we can also look, I showed you earlier what we refer to as a mean variance. We can also then analyze this by comparing the mean value and the variance value as well. So again, in a perfect world, we would be looking at the left top, where we had a very high mean value and we had a relatively low variance. Now here it is quite likely that most of you will select scenario number 4, which gives us a high mean value and relatively low variance. That's the best scenario, but this is the analysis for yield only. As I said earlier, we have a very simple way to look at the economic analysis as well. So we can look at economic analysis. So we have in the background a price file. And again, I don't really have much time to explain this price file to you, but here we account for the cost of production. So we account for a price of fertilizer using some older prices. Looking at a price also for grain yield. And now we're looking at what we refer to as the monetary returns per hectare. Looking at the same 6 box plot. But now if you look very carefully on the y-axis, where we have money per hectare, we actually have some negative values. So you can see that for certain scenarios, a farmer is actually losing money, again, especially for the low input environment. And we can also see again for our high yielding responses, now again that the returns for that particular scenario vary a lot, all the way from about 1,800 dollars per hectare down to about $150 per hectare. Now it's again quite challenging to decide what would be the best scenario to select here. So again, here if we look at the mean variance plot, things are going to be looking a little bit different. So I use this quite often in my, again, in the workshops. And again, I don't really have access to a poll here. But again, we're looking here at a variance on the x-axis. So again, we're looking at something which is a low variance and a high mean value. But depending on how risk averse you are as a person who's listening to this seminar, if you don't like to take many risks, it's highly likely that you're going to look for a scenario which is sitting somewhere over here, where your variance is relatively low and your mean value is quite high. Well, if you're willing to take a little bit more risk, if you're a risk taker, it's highly likely that you might select scenario number 4, where the variance is higher, but also your mean return value is higher as well. So normally when we take a poll, we can see that some people select scenario number 3 and some people select scenario number 4. So this goes back to one of the slides I showed you at the end, where there's the challenges with respect to the human decision-making process. We don't really know if you're a risk taker or if you're willing, somewhat risk averse. So it is again up to the decision maker to decide which of these scenarios you're going to select. But we tried as an agronomist or as an economist or as an agricultural scientist using these decision support tools to provide the stakeholder with the best management information possible. But this is then one of the applications we can look at with respect to economic analysis, going from yield to economics analysis. So I wanted to go back then to the title of my my seminar as well. So one of the other questions you might ask is, how is my variety going to perform in a new environment? So what I've selected here is a completely new environment. This happened to be Chiang Mai, Thailand, in the northwestern part of Thailand. What I'm interested in is how well will maize produce in that environment fairly high nitrogen input, 160 kgs of nitrogen. And we compare different hybrids. So we have a short season hybrid and we have various other hybrids. These are quite old hybrids. So again, this is a hypothetical scenario. So again, I'm going to look at the performance of different hybrids for this particular environment using, again, long-term historical weather data. So again, we have 7 treatments. We have 30 years of weather data. So we can easily, again, run the simulation quite quickly in a what-if type scenario. So again, I go back to the analysis. So again, then I can look at the performance, again, for instance, for harvestable yield for this location. Again, using this box plot. Again, it's somewhat challenging because we have people are tended to look at the mean or the median value. Again, we've got various types of varieties which look very similar. Maybe number 7 or number 2 is very similar. But again, the variability associated with these simulations is quite high as well. But again, this then allows us to roughly to see which of these varieties might perform well in this this particular location. I didn't have the economic analysis for this, but you can also again analyze this by mean variance. So we can plot this with variance on the x-axis and mean on the y-axis. So in this particular case, potentially that may be scenario number 7 gives us the highest mean value and also the relatively lowest variance as well. So again, the decision maker ultimately needs to decide how he wants to take this information forward. Okay, so this is some seasonal single season analysis. I think there was also a question with respect to crop rotations. Again, I don't really have time to look at crop rotations, but the DSSAT system actually also allows you to define different crop rotations, long-term rotations. So for instance, right here, I have an example. This is a real experiment looking at a wheat cotton rotation from Texas, where they're looking at wheat as a cover crop to see what will make a difference for the cotton rotation. And we also got some hypothetical scenarios in this crop rotation as well. Then I mentioned earlier that we also now have the climate change applications. So in many cases, people run climate change applications where they link the GCM outcomes, general circulation models in the crop models. I don't really have that information available, but we can quickly do, in this case, sensitivity analysis. So what I have here is, again, a hypothetical scenarios. There's actually wheat production in Kazakhstan. This is a rain-fed production system, relatively low nitrogen input. We're looking at the hypothetical response of different CO2 concentration on a low input system and a high input system. So again, this is a rain-fed conditions. And we have access to long-term historical weather data for this particular location in Kazakhstan. So again, these simulations go quite quickly. I have 10 treatments here. I have 30 years of historical weather data. So there's about 300 simulations. Again, we can go back and do a very similar analysis of what I showed you a minute ago. Again, a very simple version of the box and whisker plots. Have a look at the harvestable yield. So actually we see a tremendous amount of variability. First of all, you can see that these wheat yield levels are very low. For those who are familiar with wheat, it is only about 1 ton per hectare. So actually, increase of CO2 didn't make much difference in the low and the high ending environments, especially when we have low yields. But actually, when we have more rainfall, then we can see that CO2 does make a significant difference as well. So we've seen, especially for low input system, if you look at response of climate change, in many cases, low nitrogen or rain-fed condition might be more dominant than the actual impact of increased CO2 levels or increased levels of temperature. I have about a couple more minutes left, so I want to show you 2 more examples here. So we have various tools available in DSSAT as well. I mentioned that in my first couple of slides. Allows you to enter crop management data, enter weather data and soil data. But also want to show you a new tool that we recently added. It's called sensitivity analysis. So this allows us in a more simple way look at sensitivity of particular input coefficients for the crop model. So now I'm selecting soybean, and what I want to do is look at an experiment conducted in Florida. So I'm going to look at rain-fed conditions for soybean. Again, it's a very old experiment, 7801. And here again, my question would be quickly, how will a particular cultivar, in this case maturity group, perform for conditions in Gainesville, Florida? So this is very similar to the seasonal analysis I ran earlier, but in this case, I'm only looking at one set of weather data. So what you see here is a set of varieties we already parameterize for soybean in the DSSAT model. So if you download DSSAT, you're going to find the same type of varieties. So what I'm going to do here is select all the maturity groups as input to actually hopefully demonstrate to you that probably there's a particular variety, maturity group that performs well in the production system in Gainesville, Florida for rain-fed soybeans. So what I want to show you here is just a yield. So again, we have triple zeros for those of you not familiar with soybeans. So soybean is a crop extremely sensitive to photoperiod. So triple zeros is maturity groups that are not sensitive to photoperiod. And 10 is a maturity groups that are extremely sensitive to photoperiod. So in triple zeros would normally be grown in Canada or northern United States, while the 10s and the 9s would be grown in conditions such as Costa Rica and Venezuela. And we normally grow maturity group 7s here in Gainesville, Florida. So you can see here the actual response using one year of weather data for this particular case. And you can see response of different maturity groups. This is maturity group 10. To these inputs for the particular location. So sure. So in this particular case, surprisingly, maturity group 3 is performing better. But again, this is rain-fed conditions. So what might be happening is that early during the season, we had no drought conditions. So depending on the planting date, and again, the planting date controls photoperiod, this maturity group happened to perform better. But in normal conditions, we'll find that maturity group 7 outperforms any of these other conditions in Florida. So we can also do a similar sensitivity with each of these genetic coefficients I talked about earlier, but because we're running out of time, I want to leave a few minutes open for questions I will stop here. And again, I will encourage you, for those who have not downloaded DSSAT, to go to the website, request a copy, and then start looking around a little bit with the software, because there's many potential capabilities to really look at the genotype by environment, by management, interaction, and even also looking at economics as well, as I showed you a minute ago. So with that, I'll be glad to answer any final questions in the chat.
Great, thank you. So there's one that's recently put in. It says, for any of the crops that are not available in the software now, will the tool be able to simulate a general model without specifying a crop?
No. In order to really simulate a crop, you really need to define properly the temperature response of that crop for development, for growth, for photosynthesis. You need to define the plant composition. So these crop simulation models are quite detailed. These are not statistical models. These are based on partial differential equations. So you cannot just simulate any crop. You first need to define properly the crop genetics in order to be able to simulate it. But having said that, we are working with other people. If you have a particular crop in mind you would like to see added to DSSAT, then please contact me privately. And then if resources are available with respect to manpower and especially good data, then we can add new crops to the DSSAT system, except for the tricky tree fruit question that was posed earlier.
Great. And then there's another question recently. May you quickly demonstrate how to introduce new cultivars in the model?
I'd rather not do that on a public seminar. So basically, I mean, I can state how to do it, but you can access this cultivar file through the DSSAT portal, and you can copy and paste an existing cultivar into your cultivar file, and then use that as a template for a new cultivar. Make sure that you give it a new name and a new cultivar ID. So, but that's really part more of a hands-on workshop. I'd rather not prefer to do this in a public seminar, sorry.
Okay, and then there's a question asking how the software can be used for investigating crop rotations.
I think I already mentioned that earlier. We have a crop sequence crop rotation option in the applications. So you just need to set up your particular management, and then you can do a 30-year, 40-year, 50-year rotation. You can see how yields would change over time. You can see how soil organic carbon, so soil organic nitrogen will change over time.
Okay, thank you. There's a couple questions asking about funding for early career researchers and training. Is there any funding available for this type of work?
Not through us, unfortunately, because we're always looking ourselves for funding. I mean, if you're interested in crop modeling per se, then unfortunately you need to go back to the funding agencies and see if they're willing to support a small or large grant proposal. I know in the United States it's quite challenging. If you're participating overseas, in some cases there are quite a few NGOs will provide financial support for capacity building and training. As an example, for instance, I'm working with scientists from Pakistan who obtain support from something called the Higher Education Commission, who spent 6 months here at University of Florida in my group to work with them on training them how to use DSSAT and how to apply DSSAT in their dissertation research. So, and there are other funding agencies available as well that especially for capacity and building, they can provide small grants.
Okay, and then one, I think, quick question. Is the software able to show or predict the trend of atmospheric gases exchanged during the growing season?
So we predict greenhouse gas emissions on a daily basis now, but that's mainly the soil processes. We predict photosynthesis, so you can look at the rate of photosynthesis, but we don't actually predict how much CO2 is extracted from the atmosphere, if that's what you're looking at. Looking at the outcomes of photosynthesis, not as what goes into the photosynthesis.
All right, well, it is noon, so let's be respectful of everyone's time and we'll end it here. Sorry to those who had questions that we weren't able to answer. A lot of great participation. Thank you all for attending and your wonderful questions. And thank you, Dr. Hoogenboom, for leading this great workshop. And again, a recording will be made available on the AG2PI.org website within a few days.
So again, if you have any questions, please contact me through my private email, gerrit@ufl.edu, and I'll be glad to answer your questions.
All right, thank you, everyone.